Swayam Prabha. Digital India. Educated India.
Thank you. Right, next, the last paper of today is also presented online. These are the works done by some scholars from Center for Process Integration in the UK, University of Manchester. We have Mr. Pan, I believe, who is with us online. I visited your place last month. Did a viva for one of your colleagues there. Yeah. Right.
Okay,
today, your
screen.
presentation of Pan will be on the topic of molecular substitutes description for the estimation of pure component properties.
Okay, thank you. So let me start. Hi, everyone. Really glad to have the opportunity to present my research at the PSC Asia 2022. I'm Chung Pan from the Center for Process Integration, the University of Manchester. Today, the work I would like to share is about automatic creation of molecular substructure descriptors for the estimation of pure component properties. Firstly, in today's presentation, I would like to introduce my research in the following sequence, starting from an introduction of the background and the current status of this research area to the proposed methodologies that are addressing the research problems. The proposed method was then evaluated using a case study. Finally, a brief summary of this work. As is well known, molecular properties are of great significance in chemical process design and product specifications. For example, in separation processes, such as distillation, mixtures are separated according to their boiling points. In chemical reactions, thermodynamic properties determines reaction path and conversion rate, or in product blending, in order to maximize profit while also meet product regulations. For example, the octane number of gasoline should be precisely controlled while blending. Generally, properties of mixtures are determined by pure substances it contains. Therefore, estimating properties of pure substances is a priority. As for properties of pure compounds, for instance, normal boiling points, critical temperature, pressure, and volume, etc., are frequently used. Because of the importance of pure component properties, great efforts have been devoted into method development of molecular property prediction. From the 1950s, with limited understanding of molecular structure, various correlation methods by correlating atom number with properties have been developed in pure component property prediction. But the method is no longer suitable for complex molecules with heteroatoms such as oxygenates. Another widely used method is the group contribution method. The method is by defining hundreds of structure fragments. Then one molecule can be fragmented into a few groups. For example, in this figure, the molecule is fragmented into a few CH groups and hydroxyl groups. Basically, each group has a contribution to the property. However, their article did not show how to fragment molecules into groups. Also, groups are predefined, which is not easily adaptable to molecules. And prediction accuracy is limited while molecular structure fragments cannot be described by the defined groups. Different to the group contribution groups, which assume that each group contributes independently of other groups in the structure. Many topological indices are a function of the entire graph and thus reflect the fundamental nature of the molecular structure. One important class of topological indices called connectivity indices considers how graph is connected. The QSPR method correlates properties with molecular descriptors derived from molecular characteristics. Technically, molecular descriptors for QSPR models can be derived from group contribution groups, topological indices, and other defined descriptors. The major problem is that interpretation of the developed descriptors can be weak, provisional, or utterly lacking because some of the descriptors are chemically meaningless. Recently, machine learning methods, especially graph neural network, have been used to predict the structure property relationships of molecular. Generally utilize a graph representation of molecular where atoms correspond to knots and bonds to edges, containing information about the molecular structure. However, the convolution process of generation may result in the loss of information, making it difficult to trace the substructure's contribution to the property. Enlightened by the above methods, we try to combine the advantages of the above techniques, the automatic generation of descriptors to represent molecular structure information. In addition, the process also meets the popular data-driven concept. We only need to change the database for different properties such as normal boiling points, critical pressure, and etc. Then the property prediction model is automatically developed without intervention, which significantly simplifies the whole process compared to other methods. The simplified molecular input line entry system, the SMILES, is broadly used in cheminformatics. It represents structures as a linear string of characters. It is human readable, and also as long as we draw a structure, we can get the SMILES code. Many machine learning models have been developed using SMILES as input, such as the machine learning-based bone dissociation answer pre-calculation model based on popular informatic tool RDKit. We developed a program code to automatically extract atoms and bond collection information from the canonical SMILES code of a molecule. Representation of a molecular structure is the premise of feature generation. A molecular structure is often described using chemical graphic approaches. In this work, the concept of connectivity matrix is employed to represent a molecular structure. Here we represent the connectivity matrix of orthoxylene. Atoms in the molecules are numbered at first because there are 18 atoms in the molecule. So a matrix with 18 rows and 18 columns were generated. A diagonal entry of the matrix is used to store atomic number of an atom in the molecule with the index of the diagonal element consistent with the ID number of the atom. An off-diagonal entry is used to store bond information between atom I and atom J. If there is a bond connecting atom I and atom J, then the entry I and J are set to 0, and 1 for single bond, 2 for double bond, and 1.5 for aromatic bond. For instance, if we want to generate first-order submatrix of atom 1 in orthoxylene, we first traverse atoms that are directly connected to atom 1, which are atom 0, 2, and 6. The intersection of row 0, 1, 2, 6, and column 0, 1, 2, 6 of the matrix is extracted as a submatrix. To generate the second-order submatrix, two adjacent atoms directly connected to a bond in the molecule are considered as a single entity. The entities in the intersection of these corresponding rows and columns are used to construct the second-order submatrix of a bond. Also, by considering 3 contiguous atoms, third-order submatrix is generated. In this way, submatrix representing every atom and bond can be automatically created. Here we present all first-order submatrices. Also, from the molecular structure, we can find that atom 0 and 7 have the same environment, atom 1 and 6 have the same environment, and atom 2, 3, 4, 5 have the same environment. Although eigenvalues and the determinant are two essential characteristics of a matrix, we find that different matrices representing distinct atomic environment could result in the same determinant, leading to the difficulty in differentiating such matrices. However, such a situation is not observed for the eigenvalues of the matrices, as they are usually different when the atomic environment is different. Therefore, eigenvalues are selected for the characterization of the submatrix generated. As determinant value has a simpler form, it is transformed into a string and used as an identifier of the submatrix that is characterized by its eigenvalues. One of the advantages of the proposed method is that each feature can be tracked back to the substructure using the stored submatrix. Structure fragment can be restored. The reason why we need higher order features is presented in this table. When only use first order features, the 3 xylene isomers cannot be differentiated, while considering second order features, stereometric and paraxylene can be differentiated. Only if third-order features are considered, the three Ziding isomers are finally differentiated. Then various regression models can be used to establish the relationship between the input and output data, representing independent and dependent variables. Consequently, the AM model is used to learn the relationship between substructure features and molecular properties. In this case study, we adopted the largest normal boiling point database to evaluate our method. Of 5,276 molecules were input to generate substructure features, and Pearson's correlation method and the principal component analysis method are applied to reduce the dimension of the desired features. The AM model is constructed to determine the relationship between input and output layer. The k-fold cross-validation method is implemented to make full use of the dataset for training and validation and investigate perturbation of the model prediction performance by input features. 15% data points in the dataset are randomly extracted and used as the test set, whilst the remaining data points are randomly separated into 6 folds. At each iteration, the data point in a fold are selected as a validation set to monitor the training hypothesis, while the data point in the remaining 5 folds are combined as the training set. The test set is used to compare the performance of the trained model generated at each iteration. The detailed root mean squared error and mean absolute percentage error values for submatrices features generated using the 6-fold cross-validation method are presented in this table. The average root mean squared error on training set, validation set, and test set are 24, 37.9, and 31, respectively, whereas mean absolute percentage error are 2.54% and 3.72 and 4.02%, respectively. Root mean squared error on the test set is quite close to that on the validation set. The parity plot of and the distribution of the relative errors between the predicted and experimental properties values at iteration 5 in this table are illustrated in this figure. It can be observed that most of the 792 data points in the test set distributed along the diagonal line indicate that the predicted values are close to the experimental values. The relative error distribution demonstrates that a data point with different relative errors distributed evenly on both sides of the IE equals to 0. Specifically, the number of data points with the predicted relative error between minus 2.5% to 0 and 0 to 2.5 are 265 and 217 respectively. In addition, one recently published article developed a GPR model, which means Gaussian process regression model on group contribution features. We copied their group contribution model and we copied their Gaussian process regression model and input our submatrix features into their model. This result indicates that the proposed framework generated a better prediction result by 11.7% in comparison to the GC group contribution method when the GPR model is employed. On the other hand, we evaluated the artificial neural network model performance built upon the group contribution features and our submatrix features. Root mean squared error on the test set from the submatrix features model is 31, which is reduced by 23% compared to use the group contribution features. To summarize briefly, in this work, we proposed a novel connectivity matrix-based framework coupled with machine learning techniques for automatically creating molecular structural features of molecules. The computational result demonstrates the proposed framework is capable of generating molecular structure features. It is also found that the proposed framework outperforms the existing group contribution method. Thanks for your attention. That's all my presentation. Thank you.
Thank you, Speaker. Thank you, Mr. Pan. I'd like to ask if, well, you have about 1-2 minutes. We can have 1 or 2 short questions.
Okay.
Professor Wong.
Thank you, Mr. Pan, for a very interesting talk. My question is, in your matrix, there are actually types of atoms or types of bonds or positions of atoms, okay? They are,
Yes.
they are categorical variable actually. And you are using eigenvalues to create high order features. Is it a reasonable approach, or should we actually project it to some, transform the category variable to some continuous variable before we try to do the high order estimations?
Yes, that's an interesting question. Actually, we tried eigenvalues and determinant, but we found that because for different, as we can see here, for atom 0 and atom 7, actually they have the same environment, but the matrix are quite different because the location of each element is changed. So we found that while transform it to eigenvalues, they can be matched to the same features. But I'm not sure if we try another like projection can result in this kind of effect.
Thank you. I would make an advertisement that I'll present a very similar work tomorrow.
Okay.
Maybe we can discuss further. Okay.
Okay. Thank you. Thank you.
Thanks, Professor Wong. We have a second question.
Hi, this is Mandakini. I'm from Pfizer, and very interesting work. Thank you. Just wondering, like, if we have a very big molecule like we come across in pharma, like, how do you think there are like lots of challenges, because as the atoms will increase in number, your matrix would go like
Yes,
in, like,
that's true.
large dimension.
Yes, that's true.
So can you highlight the
yeah.
challenges?
Yeah. Yes, because as the atoms in the molecular increases, the matrix size should be increased dramatically, and the derived different orders matrix can increase. Actually, for this case study, we adopted the largest non-boiling point database, more than 5,000 molecules in it. And there is another problem, because for large molecules, I think that because for large molecules, I think there is a lack of molecular, like the experimental molecular property values. So so that's the problem why prediction of the large molecules can be challenged, because in the experimental database, we don't have such experimental data of the large molecules. But I think this method can be used to predict the properties of large molecules, but the accuracy should be influenced.
Thank you, Mr. Pan. I believe we addressed your questions. So, ladies and gentlemen, that's end of the sessions today, and I will see you later. Right, thank you.